Well, it's a great honor to be invited to speak to such esteemed and prestigious hackers as yourselves. I'm a hacker too, but I hack DNA. So I call this talk Forbidden Fruit. Yeah. I work at Harvard Medical School in Boston and MIT Biology in Cambridge. There's a big field called bioart, and many bioartists consider me a tool of the state because I aestheticize molecular biology and dangerous biotechnology. But I was co-opted. They used my one of my artworks as a poster for their conference in Winnipeg earlier this month. And they use terms like toxic life, toxicity, socio-political, ecological landscape, ethical relationships, biopolitical and cultural biotechnology, poisoning, infiltration of biotechnology. So they're aestheticizing something. There's concerns about new industrial biology tailored to produce pharmaceuticals, industrial chemicals, and the like, but also a lot of this concern is centered on crops introduced into the food supply. Now, we've been told what we should and should not eat since we've been very small, I'm sure. And in the laboratory we have restrictions. We're not supposed to eat, drink, smoke. It's a kind of, molecular biology is itself a kind of dietary regime. But aversions to selected foods is nothing new. It's actually the hallmark of human civilization, dietary restrictions. We avoid foods that we consider sacred and profane. But some of us are genetically predisposed to dietary restrictions because of severe allergies or because we lack certain enzymes. The disaccharide sugar in milk is one of them. If you're not of European ancestry, usually you can't digest lactose. Celiac disease is another example, a combination of two proteins present in wheat, barley, and rye cause an autoimmune disorder in people who are allergic to glutenated foods. Seafood allergies make up including scaly fish, are the most common potentially life-threatening allergies, at least in the United States. Peanut allergies, on the other hand, only affect 0.4 to 0.6% of the population, and recent studies suggest that the huge response to peanut allergies and all of the measures that we've taken to avoid peanut allergies are actually mass psychogenic illness. Bulimia nervosa and anorexia nervosa are clinically diagnosed pathologies. They're eating disorders, but they come from body image. And so we wonder, where this notion of desirable body image comes from. The ideal self-image is constructed by whom? And so maybe we should consider Surgeon General's health warnings on Vogue and GQ. That, like, if you read this magazine, you may end up with an eating disorder. On the other hand, obesity, even though it's not officially a disease, it's considered a serious public health problem. It's connected with type 2 diabetes, sleep apnea, heart disease, osteoarthritis, high blood pressure, high blood cholesterol, and certain types of cancer. But there are eating disorders that aren't caused by allergies or lack of enzymes, and they're caused by psychological conditions that are really paralyzing fear of certain foods. Electorophobia is fear of chicken. Acerophobia is fear of sour tasting stuff. Alliumphobia is fear of garlic. Mycophobia is fear of mushrooms. Carnophobia, not to be confused with vegetarianism, is fear of meat. And this one, I'm not even going to try to pronounce. It's fear of stuff that sticks to the roof of your mouth, like peanut butter and marshmallow whip. People really have problems with this stuff now. Gumophobia is fear of flavor. You can only eat plain rice and plain pasta. And this one is fear of chopsticks. There is, in fact, a fear of vegetables, lacanophobia. There's fear of fish, ichthyophobia. There's fear of cooking. You can only eat carryout. There's ostraconophobia, that's fear of shellfish. That's my original picture. And there's fear of swallowing. There's fear of hot foods, thermophobia. And of course, there's fear of fast foods, but this is not, so far, fear of fast foods has not been clinically diagnosed as a psychopathological disorder. We are, however, concerned about mass-produced foods, chemical preservatives, pesticides, vegetable oils, trans fats, MSG, high fructose corn syrup, and refined white sugar. Yes. From a global perspective, we could consider vegetarianism as a religious practice, but often in the Western world, it can instead be described as a culturally inspired eating disorder. They're good. We also have to consider starvation, culturally inspired. Hunger is a culturally inspired eating disorder right now and historically. In places like Palestine and Syria, hunger is going on right now. In Darfur, in Central Africa. A sign of the times in Cambridge, Massachusetts, where Harvard is and where MIT is, all of the regular grocery stores, most of them were Johnny's Food Masters, were bought out by a very famous organic food chain. And this famous organic food chain is an extremely lucrative multinational industry with skillful marketing practices and management strategies and powerful political lobby. Actually, even though we don't generally realize it, it is possible now to learn how to run a multinational corporation and still go to heaven. I was just in heaven. One of the targets of the multinational organic food lobby is Bacillus thuringiensis, the name comes from Thuringia, Germany, and it's a bacillus that was discovered there some time ago. Genes from this bacteria are given to crops, and bugs don't eat them. Now, they've been implicated in the death of monarch butterflies, but see, insecticides kill monarch butterflies too, and if you don't use BT vegetables, then you have to pour a lot of nasty stuff in the soil. And the studies that linked genetically modified corn with rats were retracted at the beginning of this month. There's no evidence to show that the BT genes in corn is dangerous to anybody, rats or human beings. And for that reason, organic farmers use Bacillus thuringiensis. It's one of these little donuts. These are like compressed BT spores of the bacterium. And it's one of the weapons that organic farmers have against pests. They put one of these donuts in the water and then they spray it all over the vegetables, and bugs don't eat the organic vegetables because Bacillus thuringiensis is an organic bacteria, I guess. And you can wash your organic vegetables, but you can't. There's no way that you can get all of the bacteria off of your organic vegetables. And the fact is, ladies and gentlemen, you eat more Bacillus thuringiensis genes eating organic vegetables than you do eating Monsanto corn. I know you don't want to hear this, but I don't think Monsanto is a sweet corporation. Efforts for robust crops resistant to pest, disease, and climate change notwithstanding, World Agribusiness is answering some pretty serious accusations about short-sighted, profit-driven practices such as risky international monoculture in world agribusiness and food crops, the propriety of the genes of rainforest species, and in some cases human populations. All of these things are highly questionable and deserve criticism. But just for contrast, let's take a look at the really dangerous poisons that are available in your local organic grocery store. Don't ever let your broccoli turn yellow in the refrigerator, because those are the flowering seed pods of broccoli. They produce cyanide-inducing glycoside called glucosinolates, and these turn into cyanide in your stomach, and in digestive juices convert that into thiocyanate, which interferes with iodine transport, and so they're called goitrogenic. I mean, that is, they can cause goiters. But it's not just broccoli. It's cabbage and turnips and don't ever put raw lima beans on your salad. There's a cyanogenic glucoside called linamarin that has the same problem, and other linamarin containing vegetables include cassava root, bamboo sprouts, and sweet potatoes. This is the cyanogenic glucoside structure. I love Holunder. I learned to like Holunder in Germany, but elderberries, of course, are also extremely poisonous unless you cook them. In the organic grocery store, there are these long rows of dry goods that you can buy in bulk, right? Like lima beans, kidney beans, navy beans. Well, let me tell you, as few as 5 raw organic kidney beans can kill you. So in those bins, there's enough in the organic grocery store. If you were to feed them to people raw, you could kill a whole city. That's the stuff, phytohemagglutinin. And of course, we've all heard about killer tomatoes. Green tomatoes, in fact, the whole tomato plant except for the ripe red fruit, contain tomatine, which is another toxic glycoalkaloid. People have actually consumed enough green tomatoes to die, and the symptoms include vomiting, diarrhea, increased heart rate with subsequent slowing of same, respiratory problems, cardiac arrhythmias, cardiac arrest, and death. They also contain, green tomatoes and green potatoes also contain atropine, another dangerous alkaloid. The green and sprouting potatoes contain substances called solanine and chalconine. These substances are not destroyed by cooking, and they're found in other plants that are members of the nightshade family, including paprika, eggplant, peppers. You scared yet? Watch out for parsnips. The skin of parsnips, especially where the plant has been damaged or bruised, it contain substances called furocormorins, and these cause phytophotodermatitis. There's an example of if you just, when you always peel your parsnips and your carrots, but don't stick your hands in the peels. Now, mushrooms. A single commonly available button mushroom, white button mushroom, contains about 300 milligrams, or 3/10 of a gram, of agarotine, which is a member of a family of toxins of volatile carcinogens and rocket fuels called hydrazines. We use hydrazines on space shuttle to push it around. All mushrooms contain this. Only 30% of these hydrazines are destroyed by cooking. So if you decide to eat mushrooms, don't eat them raw and don't eat a whole lot of them. Shiitake is associated with shiitake mushroom dermatitis, flagellate mushroom dermatitis, mushroom workers' disease, and shiitake-induced toxidermia. These are from eating raw or undercooked shiitake, and symptoms can appear after about 48 hours after eating raw shiitake and can last for about 10 days. It is a toxic and not an allergic reaction, and it's believed to be the result of the presence of a starch-like polysaccharide called lentinan. But one of the more interesting aspects of shiitake mushrooms is that they naturally produce formaldehyde. This formaldehyde is destroyed by cooking. And apple seeds and pear seeds and plum contain small amounts of a substance called amygdalin, which is another cyanide containing cyanide-inducing substance. And so if you make applesauce, don't grind up your apple cores in your applesauce. 1 cup of apple seeds, an amount equal to 1 cup of apple seeds is known to cause death. Have you ever wondered why you've never seen a cashew shell? The reason why you've never seen a cashew shell is that they're burned in the field where they pick the cashews, and if you stand downwind from the fire, you can die. Cashews are in the family of plants that include poison ivy, poison oak, and poison sumac. And they have this really nasty substance in the cashew seed itself, this cashew shell. It's like this honeycomb stuff that contains this nasty liquid called erythanol, and it is. So they are soaked, washed, steamed, boiled, broiled, all these processes to take away the erythanol from this from cashew cells. And if you are allergic to poison ivy and you happen somehow to eat a raw cashew, well, you can die. Mangoes are another plant in the same family, poison oak, poison sumac, poison oak. And actually, a few weeks ago in Cambridge, I know someone who got dermatitis from mango peels. The mango fruit is good, but the mango peels are toxic. And the toxin in alfalfa sprouts is especially insidious. It's called cannabinine. It's an amino acid, and it's an amino acid that's very much like an amino acid that we use, that's in the so-called universal genetic code. That amino acid is arginine. And what happens is the body incorporates cannabinine in the place of arginine, and so you make all these aberrant, structurally unusual proteins, and it causes an autoimmune disorder in human beings similar to lupus. Okay. And if you juice your alfalfa sprouts, you concentrate the cannabinine. Don't juice your alfalfa sprouts. Even lettuce contains toxins, flavonols. They contain, let's see, natural phenolics, glycosides, to protect it against pests. They're found in many plants. If you eat a really huge amount of lettuce, and some lettuce farm workers in California actually consumed enough lettuce to get sick. So if you eat a whole field of lettuce, you probably will get sick. Now, Have I convinced you? Hungry? There are alternatives. Play-Doh, I mean, little kids eat Play-Doh, but Play-Doh contains borax, which in Europe is on a restricted list. In the United States, it's like permitted somehow, but it's not good to eat. You shouldn't eat Play-Doh. There is a psychopathology. There is a psychopathology that is fear of all food. And it's not to be confused with anorexia. It's called sebophobia. Fear of food. But now, the most dangerous, at least mythically, the most poisonous food of all is the tree of knowledge. So if you find yourself around a tree of knowledge, you shouldn't eat, drink, smoke, or... It's not just the tree of knowledge per se, it's the tree of knowledge of good and evil. It's the forbidden fruit. It's knowledge of the best and the most heinous, abominable parts of the human soul. This is the poison. And this battle, this terrible battle between wisdom and knowledge that we've undertaken throughout human history that's still going on, we're chopping each other to pieces over it right now as I speak. This, of course, has left us less than divine. Jumping ahead of myself. But this story is much older than Judeo-Christianity. The snake in the tree. Inanna, the Babylonian fertility goddess, Ishtar of Babylon, Sumerian Inanna. Ishtar is the namesake of Easter. She's why we have Easter eggs and Easter bunnies and flowers and other fertility symbols. She planted a tree guarded by snakes, and the tree contained the tablets of destiny and a female demon called Lilith that during the Babylonian exile was incorporated into Judaic scripture. Lilith. And she was, Michelangelo included her. Notice the serpent in the Sistine has mammaries. She's Lilith. The Norse Germanic Yggdrasil, the tree of life, was also guarded by a serpent. So I work at Harvard Medical School, where at this small part of Boston where in the short period between 98 and 2003, over 35,000 articles in 2,000 journals by 200,000 authors originated in this group of buildings. It's knowledge. So George Church, the director of my laboratory at Harvard, came to me one day and tapped me on the shoulder and he said, "Joe, your name came up at faculty meeting. We want to know why there's not a lot of art at Harvard Medical School. We know you have a lot of art over at MIT Biology. What are you going to do about this?" So we have this big atrium that was originally supposed to be a Japanese garden, but they ran out of money, and instead we have these ugly green picnic tables. So I had this notion that since this is such a center of knowledge that I could, and there's not enough light or water, whatever, the conditions aren't right to actually grow anything there, but I could put the genetic signature of the tree of knowledge. I knew that one of the domestic apples, Golden Delicious, had been sequenced, and that I could put on one side, I could put binary code that relates to DNA triplets. On the other side, I could put digital number that corresponds to its position in the code. But looking into it, I found that it had recently been shown that an apple, a wild apple, a dicot, a diploid apple, from meaning that there's only 2 copies of its DNA, from mountains, a mountain range in Central Asia, is truly the ancestor of all domestic apples, and this has only recently been shown to be the case. It's called Malus cerevisiae. It's resistant to diseases that all of the domestic apples are susceptible to. It has a much wider genetic diversity than all of the 75,000 varieties of apples that exist in the world, domestic apples, because we remove genetic diversity when we domesticate plants. So we expect to find genes. We're sequencing this apple now. I got tissue. Let's see. And seeds. It's expected, we expect, since it's a smaller genome than domestic apples, which have many copies of DNA and not just 2, we expect it to be between 500 and 750 megabases, which is 750 million rungs of ladder of DNA. That's small. It's much smaller than the human genome and very small for a plant. Anyway, so with my colleague Yoav Meshar at Harvard Medical School, we used liquid nitrogen and extracted DNA from this ancient apple. We used a kind of Betty Crocker cake mix. It's called Qiagen DNeasy. We also extracted RNA, and so this is like the most closely guarded secret in paradise, right? One of the first things we did was this is a hacker machine. This is a machine that takes DNA, strands of double-stranded DNA, and sticks one end of it to a vinyl-covered slide, and then we use water tension to pull the DNA straight. So here you are looking a little. You're like some of the few human beings who have looked so deeply at this secret. But it occurred to me that 750,000, no, it's 187,000 stones in that atrium aren't enough stones to contain the genome of the apple legibly, so we could only put parts of on all these stones. And so I did this pilot project. I took the gene for Rubisco, which I've worked with in the past. It's the most abundant protein on Earth. All living things are made of protein, everything. And if you break down all living things into piles, discrete piles of proteins, then one of those piles would be really big, and it's this one, ribulose 1,5-bisphosphate carboxylase oxygenase, which is called Rubisco for short. It's an Eden protein. It's part of the Calvin cycle of photosynthesis, and when it appeared, what it does is it takes inorganic carbon from atmosphere, mixes it with sunlight, and makes food. And even though we, Homo sapiens, we don't make Rubisco, all of the complex carbon that we are created from, that we are made of, was at one time or another made by this protein, Rubisco. And so when it appeared on the once barren Earth, then came all the verdant gardens, right? And so I wanted this to to be part of the legacy I would leave on the stones. And so I mounted an exhibition at the South Dakota School of Mines and Technology in Rapid City, South Dakota, and I actually, it took me 5 days with help, and I stamped 10,000 stones with the gene for Rubisco. I figured it's going to take me like many months to stamp all the stones in that atrium. But these stones are now part of the permanent collection of the School of Mines in Rapid City. They installed it on a concrete plinth outside their chemical and biological engineering building. So meanwhile, with the help of my colleagues, I established an international consortium now consisting of over 35 investigators at more than a dozen institutions worldwide. And we are sequencing the genome and the transcriptome of this ancient apple. There's plant pathology. The apple is of such ancient lineage. These data will answer questions in evolutionary biology. The raw sequencing data comes from Karolinska Institute in Stockholm. Richard Sandberg's lab, and Josta Weinberg is also one of the principals. We're using Illumina machines, HiSeq, next generation DNA sequencing machines, and this technology is coming of age. A few years ago, it cost $1 billion to sequence a human genome. We now can sequence a human genome for $1,000. The same is true with DNA synthesis. These are some of our first data. This is the DNA run on a gel. Another close look at the ancient secret. So, meanwhile, Karolinska Institute is building this giant new building called Biomedicum. Which is going to house 5 different departments. It's part of one of the largest construction projects in Western Europe, and they commissioned sculpture. We're going to plant some of these trees, but not just the Malus cerevisiae I'm about to explain. And I designed these paper models, which I brought along with me tonight, and I should have at the podium, but I don't. That will be cast. The skin will be cast in bronze, and the flesh will be plates of stainless steel with the laser carved texts and the DNA in the inside of the peel. Actually, I'm going there for both art and science in about a week. Brigham Young University in Provo, Utah is doing the assembly. What happened? Yeah. Brigham Young University in Provo, Utah is doing the assembly of the genome. So like getting the raw sequence data is only a small part of the problem. The real hack is how to put it together in continuous pieces. And this is a real speciality of bioinformatics, and Brigham Young happens to be one of the centers of this because of their fascination with genealogy. Speaking of tool of the state, the United States Department of Agriculture, more than a dozen members of the United States Department of Agriculture are members of our consortium. They don't have the money to sequence DNA, to sequence a genome. they actually supplied the tissue, and they held a special meeting in September about our project, which I attended with my colleague, Mike Chow. This is the specimen, the tree Malus cerevisiae, where our tissues comes from. And of course, I was tempted. And I tasted it. I didn't get any smarter. Okay, many of you probably don't really know about my background in art and molecular biology. For like several decades, I've been developing, exploring ways to incorporate the human intellectual legacy in the vast, timeless, deep time terrestrial biome. My first such project really stemmed from projects in the search for extraterrestrial intelligence, where I used algorithms previously used to transmit messages to aliens to incorporate a graphic icon in the DNA of E. coli, and there's the sequence. In a later project covered in the journal Nature, it's a really interesting story. Okay, who knows what the genetic code is? Does anybody know what the genetic code is? Show me some hands. Genetic code. For those of you who don't, there are 4 different bases in DNA, 4 rungs of the ladder. And DNA, the language of genetics, is written in 3-letter words, triplets of these ladders, of these rungs of ladder. They're only, because there are 4 triplets, because there are 4 bases, there are only 64 possible triplets. And these 64 triplets correspond to 20 amino acids and only 20. And in 1957, we knew the structure of DNA, but we didn't know the connectors, which triplet went to which of the 20 amino acids. And this guy, George W. Beadle, who was at Caltech in Pasadena, was winning the Nobel Prize that year, and an expatriate German physicist, later to become the father of virology, study of viruses, and would later on in the 60s win his own Nobel Prize, Max Delbruck, decided, and his pals at Caltech decided to play a trick on George Beadle, and George went on to become the president of the University of Chicago. So they sent him a telegram, 15 seconds? Okay, I'm good. They sent him a telegram with 224 letters, A's, B's, C's, and D's, with 1 X, which was the X in Max's name. It was a clue. And it decoded in English. See, what they discovered blew them away. There's no chemical or structural connection between the amino acid and the DNA triplet that represents that amino acid. The DNA triplet has as much to do with the amino acid as the word red has to do with the phenomena of perception, or whatever, of the color itself. And what that means is that DNA, that the genetic code operates by the rules, by the laws of natural language. And that's crazy. Like, we go to the edge of the universe, and we find our own cerebral cortex imprinted on the wall. We get to the limits of knowledge, and we find our own language reflected there, and science waxed poetic. They had arguments for a couple of years about whether or not there were spaces between the words. They called it comma and comma free code. Max Delbruck thought there were spaces between the words. George thought there weren't. The 224 letter telegram had a space code on. It said, break this code or give back the Nobel Prize. It took George a couple of days. With one of his grad students, he broke the code and he sent a telegram back to Max Delbruck in Pasadena that said, yeah, I got your message just fine, also in code, but with no space. Got your message just fine if we could do the final step, because the genetic code wasn't figured out for another decade or so, the places, the matches. So George and his pals in Pasadena raced to put together a response before the end of the Nobel Prize ceremonies, and they put together 174 toothpicks of 4 different colors. And the air freight officer delivered it to George when he was delivering his formal address after the award ceremonies, and damn if he didn't decode it at the podium, and it said, "I am the riddle of life. Know me, and you will know yourself." And that, friends, is the edict of Apollo at Delphi. Know yourself, and you will know the secrets of the universe and the secrets of the gods. Unfortunately, George and Max couldn't synthesize DNA. It wasn't possible to conveniently synthesize DNA until the 1980s, and so in the 1980s and early 1990s, I synthesized Riddle of Life at Free University in Berlin and sequenced it. There we go. It was the I am the Riddle of Life molecule. And my next projects had to do with I had a girlfriend when I was a young man who illustrated a children's story about the spoiled child who could find no happiness until she met a mouse that had a map of the world in its ear. And so this was at the time the most significant advance in cartography since Gerhardus Mercator. It's the Kobe image of the Milky Way. It's the first time we were ever able to image our own galactic cores looking through the intergalactic dust clouds. And DNA is base 4, 0, 1, 2, 3. The 4 bases are 4 consecutive sizes, and so you represent them, we represent them all the time as base 4. So you can convert a binary, because this picture was made with CCD imagers and liquid helium-filled dewars in a quiet orbit, and it was spit out data, binary data, that we massaged to get this image for the human sensorium. But if we make the data directly into DNA, then you end up with long repeats of the same nucleotide, C, 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 and that the repeated electromechanical torsion on the molecule causes it to twist and curl, and so all the machinery and cells can't operate on it, and you can't sequence it. And so this was my first, one of my first DNA hacks. It was how to write language into DNA that had parity with the original data, but that was biochemically and biologically friendly. Of the 64 codons, 3 of them are stop codons. These 3, and I use those as informational statements. And because of that, I was able to put in desk drawers, like file cabinet drawers. So each one of these yellow sequences is a unique sequence for an enzyme to come cut the DNA just there. So if I had a mistake in synthesis, because this was back in the 90s, and synthesis wasn't as efficient as it is now, then I could just pull out that desk drawer, fix it, put it back together. But I had a problem, and that is I ended up with a biologically useless piece of DNA that the cell, the organism would tend to rearrange or eliminate within generations. Now, they call me the father of bioart because I made the first recombinant artwork. But the real father of bioart was this guy, Edward Steichen. He had an exhibition of his delphiniums at the Metropolitan Museum of Modern Art in 1936. He used his asthma medication and mutagenized his delphiniums and made them grow giant and bright blue, brighter than any other delphiniums. You can still buy them from Burpee Seeds. It's not as if there weren't horticultural shows or flower shows all over the world. It was just, this was the first time that such a manipulation was treated in a formal way as a work of art. Now, bioartists are still really stuck on the idea of bizarre and garish mutations. They want to make, green bunnies and furry fish and but see, I'm interested in a different kind of manipulation. I'm interested in a more ecologically friendly and more environmentally compatible form of genetic manipulation than any of the genetic manipulations of the past, including all of those of molecular biology. I just mentioned, it's like Borges' story about a map of the world that coincides with itself. I just mentioned that there were 20 amino acids and 64 places to represent them, 64 triplets to place. So some of these are redundant. And so this is based, you can change this, one of the redundant codons, and still in general, not all the time, but in general, end up with the same protein. But those instances where you cannot, we take those into account. This is called silent code. So I could write binary numbers under a gene without changing the output of the gene or changing the size of the gene. It was a way to like write a song underneath another song. But it wasn't very efficient. I ended up having to use a lot of DNA to write relatively small volumes of information, and then I got scarlet fever in Osnabrück at the Media Arts Festival drinking somebody's beer. And I mean, I had strep throat before. You get that from the same organism, streptococcus, but the symptomology of scarlet fever comes if you have to be lucky enough not only to be infected with streptococcus, but to be infected with streptococcus that is sick itself, that's infected with a virus called T12. And that's how you get, but anyway, I didn't have health insurance. I was in Germany. So I was really sick, and I got on the train for Essen, and my friends in Essen finally got me tetracycline by pretending to be me, and I pretended to be them, and I got the, but before that happened, I was delirious, and I couldn't stop thinking about math and DNA, and I know, 5, right? Yes, I couldn't stop thinking about math and DNA. And I came up with a way to put not just one layer of information under a gene, but many, many layers of information, like babushka dolls. And that worked because every DNA triplet can be represented by 3 unique numbers. One number is the DNA itself. CCG is 000011. The silent code, redundant codon binary number, in this case 11, and the base 20 number that corresponds with the amino acid that is translated to. And so then I made this mathematical genetic code, and it let me write 3 layers of information. So there's, every DNA molecule thus has 3 pages of information available. The DNA itself is one of the numbers. The protein it's translated to is one of the numbers, and then there's the redundant codons, that's the silent code. But instead, instead of writing code I wrote a map of a hypothetical but very precisely defined DNA molecule in the open spaces part. And it also has 3 pages of information available. Now, it's even more efficient than real-world DNA because I could use those codons that I couldn't use in a biological context to hold information. And so then I wrote the map of a 3rd molecule and put input data and so on and so forth. And so on and so forth. And my first, I called it DNA manifolds, and my first project was to put the writings of this guy Heraclitus. Heraclitus was the greatest scientist of his age. He wrote a great work called On Nature that all his contemporaries, raved about, but it's been totally lost. One of the laws about mathematics with triangles is named after Heraclitus, but all of his work in mathematics has been lost. Only 11 pages of fragments of the work of Heraclitus remains. And one of them, because I was stuck on Delphi, says, "The Lord's, whose oracles are at Delphi, wishes neither to conceal nor to reveal, but rather to signify." And so I wrote those into the genes of a fly. For the white eyes, it's a natural but rare mutation in fruit flies. This is George Church wearing the riddle of life. About a year ago, he put the first full-length book in DNA, and actually Microvenus, the first genetic artwork, got cited in Science, and it was the first time art journal, a journal in art, got cited in Science. And so the data of the Malus cerevisiae genome was coming in, and so I got together with my old-time collaborator, Dana Boyd, and one of my colleagues from Church Lab, Madeline Price-Ball, who had been making shrunk Wikipedias for third world applications to give versions of Wikipedia to the third world for kids who couldn't get online. So we have between a 100 megabyte and 350 megabytes. At 350 megabytes, it's the top 50,000 pages, the most 50% of the most visited pages in Wikipedia. And I guess my time is running out, but I guess you can figure out what I'm going to do. I want to make apples to tempt the devil. We get to name this apple, and George Church in Latin is ecclesia, and malus means evil. It's a pun in Latin. The reason apples get the name evil, it's lost. But this is the last slide. So Malus Ecclesia will be the name of the wikified apple. And when I got to Hamburg today, the hotel room they gave me was 666. Thank you very much, ladies and gentlemen.
Okay. Maybe just stay here.
Sorry, on time.
You're just really perfectly on time. That was great. I think we don't have really a lot of time for a Q&A. I can maybe ask some single question I have right now. Is just, is there enough redundancy in the DNA? It's just so it's still an apple. It's still the information for the apple, yes, plus the 50,000 articles of Wikipedia.
Proteins, no change in phenotype. It
So
looks like an apple, tastes like an apple.
So it's just kind of biological stegano. What's this thing called where you hide data and images? It's just a bit like that. So you just use the unused portion of the information. Yeah, you can read it. Well, it's in the other way as well, but it's both things in the same thing, the information and the... Yeah, so I don't want maybe a quick question, but I think you all kind of... I think... Thanks, Joe. It was great having you here. Okay.